Hi everyone, my name is Katharina Mauel and I was a PhD student and postdoc in the group of Professor Elvira Maas at the Limes Institute in Bonn, where I was working on splenic macrophages and infectious diseases. And I am the first author of the publication CD163 positive red pulp macrophages interact with marginal metallophilic macrophages during blood stage malaria to maintain splenic architecture. And in the next 2 minutes I will tell you all about it. So amongst the well-known and well-described splenic red pulp macrophages, we have discovered a novel heterogeneity which is based on the expression of the iron receptor CD163. And these CD163 expressing red pulp macrophages, they occupy distinct niches in the spleen where they interact with other cells and fulfill homeostatic functions. We have studied splenic macrophages in the context of blood-borne infections such as malaria. And what we have discovered is that this novel CD163 expressing red pulp macrophage population is depleted very early after the parasite infection and does not recover even long-term after the parasites are cleared. In addition, sequencing experiments have shown us that these CD163 expressing red pulp macrophages interact with other macrophage populations in the spleen to maintain structural integrity of the organ, and that this signaling and communication network is disrupted upon the infection, which is associated with the disruption of the splenic architecture. The spleen is the organ in our body that is responsible for the filtration of the blood to maintain a healthy circulation. So if there is any harmful substances or pathogens in our circulation, it's usually the macrophages in the spleen that take up those particles and degrade them. So in the context of our novel CD163 expressing red pulp macrophages, we found that they are not only influenced by a parasite infection, but also by viral or bacterial infections. And that the loss of this population, no matter what the infectious context is, leads to an impaired recovery of the splenic tissue structures and iron stores. My favorite figure is figure number 3 because it is about a new and really advantageous fate mapping mouse model that we have developed during my PhD, which allows to discriminate the developmental origin of macrophages, so whether they are yolk sac or bone marrow derived, which has a significant impact on their homeostatic or disease-related functions. And also this figure contains really beautiful and colorful immunofluorescent images. On the one hand, we would like to define the exact signaling and communication pathways between the different splenic macrophage populations, which might help to improve the recovery of the splenic architecture. And on the other hand, having identified this novel CD163 expressing macrophage population, which plays an important role in various infectious diseases like parasites, bacteria and viruses, this could be explored further in the context of future treatment strategies.